What we're about to dive into is a true landmark moment in medicine. I mean, it's a story that sounds like it's straight out of science fiction, but for one baby, it was the very real difference between life and death. This, right here, is the dawn of personalized gene editing.
Think about this for a second. The instruction manual for a human being, our DNA, is 3 billion letters long. So what happens if just one of those letters is wrong? A single, tiny typo that could be fatal. But what if? What if we now have the technology to find that exact typo and just fix it?
And you know, this isn't some hypothetical question. For a newborn baby, who we're going to call KJ, this was literally life and death. His story is right at the heart of this incredible scientific breakthrough.
So within just 48 hours of being born, KJ was lethargic, he was struggling to breathe. And the cause? A condition called CPS1 deficiency. It all boiled down to a single-letter typo in one of his genes. That tiny error completely broke a crucial process in his body that's supposed to get rid of ammonia. So instead of being flushed out, that ammonia was building up to toxic, life-threatening levels.
The stakes here, they couldn't have been higher. This condition has about a 50% mortality rate in infants. The only established cure is a full liver transplant, but babies are often just too small and fragile for that kind of major surgery before irreversible brain damage sets in. KJ was in a serious race against time.
But what if, instead of replacing the entire organ, you could just fix the broken gene inside of it? This is where a revolutionary new technology comes into the story, moving from the lab to a patient's bedside at just breathtaking speed. Okay, so to really get the breakthrough here, you need to understand two types of gene editing. You've probably heard of CRISPR-Cas9, right? Think of it like a pair of genetic scissors. It finds a spot and just snips both strands of the DNA. It's powerful for sure, but cutting the code like that can lead to unpredictable results, some unwanted errors. Now, the tool used for KJ, called base editing, is way more precise. It's less like scissors and more like a pencil with an eraser. It doesn't cut the DNA. Instead, it finds the single wrong letter and chemically rewrites it into the correct one, leaving everything else untouched. It's just an incredible upgrade in precision. So here's the key takeaway. Base editing gives us a safer, much more accurate way to fix the root cause of so many genetic diseases, those single-letter typos that scientists call point mutations. For KJ, this meant they could target the exact single C that was supposed to be a T in his DNA and simply rewrite it. Just amazing. With KJ's life hanging in the balance, a team of scientists and doctors began this unprecedented race to design, test, and deliver a unique N-of-1 therapy. That is, a medicine custom-made for just one single person, and to do it faster than ever before. This timeline, it's just staggering. From KJ's diagnosis at birth, it took only 1 month to create a cell line for testing. By month 5, all the safety studies were done, and by month 7, the first dose of this personalized medicine was actually administered. To put that in perspective, this is absolute light speed for drug development. It's almost unheard of.
And this quote just brilliantly illustrates the significance. This wasn't just a win for one patient, it was a proof of concept. It showed the entire world that personalized genetic medicine isn't some distant dream. It can be a rapid response reality for patients who desperately need it.
So after all this lightning speed development, the therapy was delivered. But the huge question remained, did it actually work? Well, let's take a look at the results. You know, the numbers really do speak for themselves. This chart shows KJ's plasma ammonia levels. That's the toxin his body couldn't clear. Before the treatment, his levels were way up in the danger zone. After he got the base editing therapy, they dropped dramatically, falling right into the normal, safe range. It was a crystal clear biological sign of success.
And this biological change translated into real-world, totally life-changing benefits. He could eat more protein, which is obviously essential for a growing baby. The dose of his daily medication was cut in half. And, maybe most critically, when he got sick with common viruses, something that would have normally triggered a life-threatening crisis, he recovered without one. The therapy was working like a metabolic safety net.
KJ's story is a massive success for treating a liver disease. But a success like this immediately raises a new, way more complex question. Can we take this victory and apply it to one of the most challenging organs in the human body, the brain? So the liver is a relatively easy target. It's basically designed to filter the blood so it easily soaks up the therapy. The brain, however, is a fortress. So what makes editing genes inside of it so much harder? Well, there are 3 main hurdles. First up, delivery. The brain is protected by something called the blood-brain barrier, a highly selective wall that keeps almost everything out, including our therapies.
Second is complexity. Fixing one enzyme in the liver is one thing. Correcting a problem inside the brain's massive, interconnected network of neurons is a whole other challenge. And finally, measurement. With KJ, a simple blood test showed his ammonia levels. How do you safely and accurately measure if gene editing is working deep inside a person's brain?
So because of these challenges, the first steps for gene editing in the brain are going to be really cautious and super targeted. Scientists think the most plausible first attempts will involve injecting the therapy into a specific, localized brain region to treat something severe, like a life-threatening form of epilepsy caused by a single-point mutation. So while the brain is a major hurdle, the technology itself is continuing to evolve at a blistering pace. The success with KJ opens the door to thinking about how we scale this, from a therapy for one to therapies for many.
The base editor used for KJ is incredibly powerful, but it can only perform 4 of the 12 possible ways one DNA letter can be swapped for another. Now get this, there's an even newer tool called prime editing that's a huge step forward. It basically acts like a true search and replace function for your DNA. It can make all 12 of those single-letter corrections, and it can even fix small chunks of missing or extra DNA. This just dramatically expands the number of genetic diseases we could potentially target. But how do we make these cures affordable? The key is standardization. The goal is to create a platform. Think of it like a factory assembly line. The delivery vehicle and the editor enzyme become standard, mass-produced parts. So for each new disease, the only thing that needs to be custom-built is one small molecule, the guide RNA. The guide RNA is like a GPS coordinate telling the editor exactly which typo to fix in that 3 billion-letter genome. By only customizing that one small part, development time and cost could plummet, making personalized medicine a reality for many. Ultimately, KJ's story is so much more than just a single data point. As the NeuroView Journal says, it's a landmark. It provides a blueprint, a source of incredible hope, and a ton of insights that will guide scientists and doctors for years and years to come.
This case proves what's possible. We now have the tools to correct the typos in the code of life. So the challenge ahead is no longer just a scientific one. It's about logistics, it's about ethics, and it's about economics. How do we take this incredible power and make sure it benefits not just one person, but everyone who needs it?